In today's lecture at Medical Sciences, we want to discuss on transport of substances across the cell membrane, or what we can call plasma membrane. And as we begin, if you're new here, please, and haven't subscribed, please go to the subscription button and make sure you subscribe, like the video, and share to many others so that we can continue to grow. So in this session, we are looking at how substances cross the cell membrane, or how are they transported across the cell membrane, either moving inside the cell or moving outside the cell. And among the types of transport, we're going to discuss about passive transport, whereby we are going to tell you that under passive, we are going to categorize them under passive transport. Can either be passive diffusion. We're going to see passive diffusion versus facilitated diffusion. And this facilitated diffusion, it can either be due to carrier proteins, whereby the carrier proteins are the ones that are responsible for enabling the transport of substances inside and outside the cell. We can also have ion channels. Ion channels, and these ion channels we are talking about can either be ungated or they can be gated. And among the gated ones, they can either be voltage gated, they can be ligand gated, they can be mechanically gated ion channels. So these are the classes we can talk about, where we have facilitated by carrier proteins and ion channels. Then under the ion channels, these guys transport cations and anions across, like sodium, like potassium, chloride, and we are saying they can either be ungated, without a gate, or gated. Then another special type of passive transport we are going to talk about is osmosis, whereby osmosis applies to movement of water via aquaporin. Then under active, we're going to look at, it is categorized into two, whereby one, we have primary. We have primary active transport and secondary active transport, whereby we shall tell you that under primary, this is whereby in this method there is hydrolysis of ATP directly, but here we utilize ATP indirectly, as we shall explain in details as we move on. And the type of secondary active transport, we are going to categorize them into three, whereby we are going to have what we call uniport, we're going to have co-transport, then we are going to have also what we call antiport. You can call it antiport or counter transport. Co-transport, you can call it symport. So these are the categorization of secondary active transport. Then finally, we shall look at macromolecules. These are large substances, whereby we are going to look at transport of large or big substances, either by endocytosis or exocytosis, or transcytosis, whereby under endocytosis, we're going to look at phagocytosis and pinocytosis. And sometimes we can look at receptor-mediated endocytosis. Receptor-mediated endocytosis. So these are the different ways how molecules can be transported across the cell membrane. So after looking at this, we are going to start with passive transport. And under passive transport, we have category 1 where we are looking at passive diffusion. But before we go at passive diffusion, we want to ask also, what is the meaning of passive? Here, passive means no ATP is involved, no energy is needed in the form of ATP, so we don't utilize energy. And among the passive transport, we are moving substances from region of their high concentration to region of low concentration. That is, so here the substances are moving along the concentration gradient from the regions of higher concentration to regions of low concentration. That's why we're saying ATP is not required. So that is what we call passive. And the one of its kind is passive diffusion, whereby passive diffusion here we see that components like lipid soluble components which are lipid soluble, and then gases like carbon dioxide, like oxygen, like nitrogen, and other lipid soluble substances, be it drugs or hormones which are steroidal, or cross the cell membrane by passive diffusion. Why? Because they are lipid in nature, and remember the cell membrane is made up of a phospholipid bilayer. And because it is made up of a phospholipid bilayer, so lipid soluble substances cross directly the cell membrane. Whereas also gases like carbon dioxide, we are going to see also gases like carbon dioxide, oxygen, nitrogen also enter freely, and other smaller molecular weight substances, smaller substances like urea which is very small, like urea can also cross free by passive diffusion. So these are the examples we can see about the substances which are transported from outside the cell inside the cell by passive diffusion, directly penetrating the cell membrane, whereby we have seen lipid soluble components, gases, and smaller substances can cross by passive diffusion along their concentration gradient until equilibrium is established. If outside they are very high, so we are seeing them moving because outside they are very high, they have higher concentration, and inside they are low concentration. So they are moving along the concentration gradient until we attain an equilibrium. So that is mechanism number 1, which we are calling passive diffusion. Then the second one is facilitated. Under facilitated diffusion, we have seen either we use a carrier protein or we are using ion channels. And the first thing we are going to look at is facilitated diffusion by use of carrier proteins, whereby the carrier proteins, in case I have a carrier protein which is across the cell, this is a carrier protein. So this protein is going to enable or to carry ions from their region of higher to region of low. So still we are following the concentration gradient and no energy is needed, but it is being enabled by a carrier protein. That is what we can talk about. So we're going to see compounds which are transported by the carrier protein. And these examples we have include, these ones you can call them, this is a carrier protein, or you can call it a transporter. They are regarded as transporters. And example is GLUT or glucose transporters. That if I have high concentration of glucose outside the cell, it is carried inside the cell, and we find the glucose inside by glucose transporters, whereby we have glucose transporter type 1 up to type 5, whereby in the liver we find GLUT2, which enable entry of glucose in the liver in the presence of insulin. So this is what we are calling carrier proteins, facilitated diffusion by carrier proteins, or what you are calling transporters, like glucose. Example is glucose transporters that enable movement of glucose from the region of higher to region of low concentration. This is what we can call by the carrier protein. So this is mechanism number 2, which we can talk about. Then among the facilitated ones, we have also ion channels. And we have said ion channels can either be ungated, whereby you have a channel which has no gate. So these are ungated. And these ones allow influx of ions into the cell from their region of higher concentration to region of low concentration. Then we have also the gated type, whereby the gated type we have either voltage gated, then we have ligand gated and mechanically gated. So we are saying under ion channels we can have ungated, and remember here we are carrying substances which are water soluble. So facilitated diffusion deals with water soluble substances. Facilitated diffusion looks at water soluble substances which cannot cross directly the cell membrane. So the substances which we are carrying by the direct diffusion, by facilitated diffusion, these are water soluble components, or they are ions, so they cannot cross the cell membrane. So the next gate we are going to discuss here is the gated ones, whereby we have those which are voltage gated. You can have voltage, they are voltage gated. How? And an example of a voltage gated channel, we have calcium channel. This is the voltage gated, whereby it is stimulated by depolarization, that when there is entry of sodium ions, inside it becomes positively charged and outside it becomes negatively charged. This is what we regard as depolarization. So when the cell becomes depolarized, it creates a power stroke which stimulates the opening of the calcium high voltage gated channel, leading to influx of calcium inside the cell. And this calcium can bring about contractions. And example of this channel, they are found presynaptic, in the presynaptic neurons, whereby their entry enables release of neurotransmitters by exocytosis. So this is calcium high voltage, calcium gated channel. So these are voltage gated. We are calling them voltage gated calcium channels. Voltage gated calcium channels, which enables release of neurotransmitters from the presynaptic neurons, hence enabling propagation of an action potential or cellular response. So this is one example of the gated type. Another one is what we call ligand gated. And those ones which are ligand gated, you can have here type of a channel. This channel we are having, it has what we call a ligand. It is gated and it has a receptor. This is its receptor. And this receptor on this gated channel is where the ligand sits. That in the presence of a ligand like a hormone, it comes and binds at the receptor. So this is a ligand, and after binding on the receptor, it leads to the opening of the gate. So the gated channel opens, allowing influx or efflux of substances. And among the types of ligand gated, these ones deal with ligands can be hormones, ligands can be drugs, that when they bind to the receptor, they are going to trigger the receptor to open, leading to influx. For example, sodium, when we have sodium, they are able to enter into the cell. So we are seeing when they open, they allow entry of sodium ions. So these are what we call ligand gated. So this was 2A, B, this is for the gated, and gated, C is the for high voltage gated calcium channels, then D is for ligand. D is for ligand gated, which these ones enable propagation of action potential at the postsynaptic neurons. For example, we see that when we release neurotransmitters by exocytosis from the presynaptic neuron, these neurotransmitters come and bind on the ligand gated. So the ligands are the neurotransmitters, which cause opening of the gated ligand gated channel, hence leading to influx of sodium ions, hence enabling propagation of an action potential. So then the last one we can talk about is mechanically gated. We have others which are mechanically gated, and these mechanically gated channels, we are calling them mechanically gated. Then these ones, they were ligand gated. Ligand gated. So mechanically gated channels, for them we are seeing they are going to be stimulated to open the gate due to presence of mechanical substances like trauma, like pressure, increase in pressure, and any other physical substance which triggers them to open. And when they open, they enable transmission of the impulse. For example, in the sensory neurons, this one we can find them in the sensor. That if I touch in fire or something that is prickly, the mechanoreceptors are stimulated by the pressure, hence causing opening, leading to influx of ions inside the cell, hence causing propagation of an action potential, and we can be able to sense that there is a change in stimulus. So these are mechanically gated channels. So as we are finishing forms of passive transport, in summary, we have looked at passive diffusion, which is movement of materials from regions of higher to lower, and we have seen it is majorly for lipid-soluble components and ionized components like gases and smaller molecular weight substances, which cross directly a phospholipid bilayer. Then we have seen under facilitated, the transport is also passive, no ATP is involved, and substances are moving from regions of higher to regions of low concentration. But the difference is that this process is using aids like carrier proteins or ion channels. And the example of the carrier proteins we have seen, we can call them transporters, like glucose transporters. Then the ion channels, we have seen they transport electrolytes which are charged, and they can either be ungated without the gate, or they can be high voltage gated, like calcium channels on presynaptic neuron. Then we have seen ligand gated ion channels which require hormone drugs or neurotransmitters to bind to cause efflux of sodium ions. And we have also seen the ones which are mechanically gated, that are triggered by physical substances like change in pressure, which causes transmission of impulses in the sensory neurons, and you can respond. So this is what we can discuss under passive transport. The next discussion is on active transport. And active transport, the word active means we are utilizing energy in the form of ATP. And this energy in the form of ATP, it is the one that drives substances from regions of low concentration to regions of high concentration. So here we are moving against the concentration gradient. We are moving against the concentration gradient, and we saw in our opening discussion that they are divided into two, whereby these ones are categorized. We said they are categorized into primary active and secondary active. And under primary, we saw that they use directly ATP. Under primary, we saw there is direct utilization of ATP, there is direct hydrolysis of ATP. And examples, we are going to see sodium stroke potassium ATPase is one of them, whereby if this is sodium stroke potassium ATPase pump, these are carrier proteins which we are going to use. So this pump, what it does inside the cell, it carries 3 sodium ions. It has what we call 3 receptors for sodium ions. Then outside it has receptors for potassium. 2 potassium bind outside the cell. So in the presence of energy, when there is provision of energy, whereby there is direct hydrolysis of ATP to form ADP plus phosphate inorganic, in the presence of ATPase enzyme, it is going to drive the 3 sodium ions on the receptor, and it spins, takes them out, then it brings the 2 potassium ions inside. So we are taking 3 sodium ions out, then we are bringing inside 2 potassium ions inside. This is the mechanism. So in the presence of ATP, energy spins this carrier protein to pump out 3 sodium ions and bring in 2 potassium ions. And this one, the significance of this pump, what we are calling the sodium stroke potassium ATPase, it enables the maintenance of resting membrane potential, and it is also responsible for maintenance of body pH. So body pH is also maintained by this mechanism whereby substances like glucose, amino acids are transported depending on the sodium stroke ATPase pump, whereby it pumps 3 sodium outside and 2 potassium inside. Why? Because inside this carrier protein we have 3 receptors for sodium, and outside we have 2 receptors. So that is active transport, primary active transport. From there we are going to look at secondary active transport, and we said under secondary, here we are using ATP indirectly. How? We are going to see that under secondary active, one component is moving along the concentration gradient, whereas the second substance is moving against. So the one which is moving against the concentration gradient depends on the one which is moving along to be taken into the cell. And we said examples of secondary active transport include, we can classify them under uniport, whereby if this is uniport, uni means 1. So this carrier protein carries one type of a substance. For example, if I have a substance glucose, this substance is moved outside, or it is taken inside the cell by the uniport. At the same time, another glucose is also taken. So it is uniport deals with the same transport of same molecules in the same direction. Same molecule, same direction. Uniport. That is one of the secondary active, whereby one is moving along the concentration gradient, the second one is moving against. Then another type is what we call symport, or you can call it antiport, or you can call it co-transport. So co-transport or symport means this is a form of secondary active transport, whereby if one substance, like maybe I have substance like sodium, when it is being, if I have a substance like sodium and is being taken inside, another substance like glucose also says I will not remain behind. You also move together in the same direction. So you bring sodium inside, at the same time glucose depends on the, whereby sodium is moving along the concentration gradient, and then glucose uses the energy to move against. So glucose is moving against, whereas sodium is moving along the concentration gradient. Another example, apart from sodium stroke glucose, we can have another one like sodium stroke iodide, symporter. We have also sodium stroke amino acid, symporter. All these are examples of a symport, or what we call a co-transport, using ATP indirectly, whereby they are depending on another substance which is moving along, and another one moves using its energy. Then another form of secondary active transport is what we call antiport, or you can call it is an antiport, or you can call it a counter transport. whereby a countertransport, anti means they cannot meet, they are moving in the opposite direction. For example, if we have a substance like sodium, and it is being moved by this carrier protein inside the cell, another substance which is different, like hydrogen ion, is pumped out via this carrier protein. So hydrogen, so they are moving in the opposite direction. Here, different substances move in opposite direction. An example is sodium stroke hydrogen co-transport. We have also another one, sodium stroke 2-chloride stroke potassium co-transport. All this, this one is the form of a co-transport. This one lies up there. Then for the antipode, we have also sodium stroke calcium. That as sodium is entering in, calcium is pumped out. So these are examples of a counter transport, or what you can call antipode. Then another example we can add here among the sympod is also sodium stroke 2-chloride stroke 1-potassium symporter. All these are the examples of symporter, or what we call co-transport, whereby substances are moving in the same, whereas antipode, they are moving in opposite directions. So this is what we can discuss under active transport, whereby we had primary active using a carrier protein, which pumps out 3 sodium and brings inside 2 potassium in the presence of ATP being hydrolyzed directly. Then for the secondary, we said they use indirectly ATP, whereby we had a unipod, whereby the same molecules moving in the same direction. Then symporter or co-transport, substances, 2 of them which are different, moving in the same direction. For example, sodium moving inside, and also glucose follows. That is what we call a co-transport. Or sodium enters and iodide says we shall move together. Or sodium enters and amino acids also enter. Different components moving in the same direction. That is sympod. Then antipode is you have 2 different substances, and these 2 different substances, one is moving along the concentration gradient, and another one moves against or in opposite direction. Another example here, you can give chloride stroke bicarbonate, what we call chloride shift. So when one is entering, another one leaves to equalize the charge. That is what we call antipode. So this is what we can see under active transport. Don't forget they are categorized into 2, primary and secondary. And the secondary ones are further categorized into 3, unipod, antipod, and symporter. So we can end this discussion of ours today by looking at transport of macromolecules. And here under macromolecules, these are the ones which are big in size, and they are either by endocytosis, exocytosis, or transcytosis. So we want to see how these movements occur in the cell. And we are beginning with endocytosis. We are looking at movement of macromolecules. So we can put it in this position. So we are seeing movement of macromolecules These macromolecules are big molecules, and we said they can enter either by endocytosis. Endocytosis, which is categorized into phagocytosis, pinocytosis, and receptor-mediated endocytosis. Receptor-mediated, whereby phagocytosis is cell-eating. This is cell-eating. Then pinocytosis is cell-drinking. And here, as receptor-mediated, we are using clarithin-coated. Clarithin-coated pits. Clarithin-coated pits. So these are different types of endocytosis. Endo means we are moving substances from outside the cell inside the cell. And for example, phagocytosis, if I have a cell, and this is the cell membrane, so what is going to happen? This cell membrane, if I have a molecule outside, and this molecule is like a bacteria, it is going to be attached on the surface of the cell. Then the cell forms what we call an invagination. Forms an invagination. And after forming the invagination, what we can say, it forms what we call a false mouth. It is going to engulf, forms a false mouth and engulfs. It engulfs the bacteria. So it goes towards the bacteria and engulfs it by forming what we call a false mouth. At the end, we shall have a cell which has engulfed the bacteria inside. This is the bacteria inside in the phagosome. After invaginating to form a false mouth, which we are calling a false mouth, it invaginates, it forms a pseudopod, or a pseudo, or what we call a false mouth, and then this false mouth engulfs the bacteria or the virus, and after engulfing it, it puts it in a vesicle whereby we call it a phagosome. Forms a phagosome, and this phagosome, which is formed inside the cell, it is going to fuse with the lysosome enzymes. These are lysosome enzymes, and when they fuse with the lysosome enzymes, they form what we call phagolysosome. And the phagolysosome which is formed, these lysosomes come and start breaking down the bacteria and destroying it. So the lysosomes, they kill or they break down the bacteria. After breaking down the bacteria, it has been destroyed. So this is what we call phagocytosis, which we are calling cell eating. Cells are eaten or engulfed by microbes. So we see white cells doing the work of phagocytosis. White blood cells do this work, and other macrophages. So another type of macromolecule is pinocytosis, which I want to take together with the receptor mediated. That if I have a cell, here, under pinocytosis, we are going to see it is cell drinking. Pinocytosis, cell drinking, whereby solutes in an extracellular matrix are going to, this one is going to form an invagination. After forming an invagination, these solutes in the extracellular fluid enter into the invaginated point, then later they come and cover. So at the end, we shall form what we call a vesicle containing solutes within the cell. So this one will be the vesicle, which is formed after invaginating. So we see solutes are taken in by pinocytosis, which are calling cell drinking, whereby it forms a fourth mouth, what is an invagination, then it enters this one, then after entering it into the cell, it stores it in a vesicle. That is what we call pinocytosis, which is cell drinking. This one is somehow different from receptor mediated, whereby under receptor mediated endocytosis, we are using clarithin pits, whereby if I have a cell, this cell is going to form a pit, and this pit has receptors. It has clarithin pits. This is a clarithin with receptors. And if I have a molecule like LDL, these can be like receptors of LDL come and bind, and after binding on the clarithin, which is receptor mediated, is going to invaginate and surrounds this cell. At the end, we shall have our LDL in the phagosome with the receptors. This LDL will be inside the cell. So this is what we are calling receptor mediated using clarithin pits, clarithin coated pits, whereby they have receptors for the substances like low density lipoprotein. And this one is the one that enables metabolism of LDL or excretion of LDL. So if someone is born without these receptors, they can never excrete or have receptors for LDL from the peripheral tissues to be excreted from the liver in the bile. So this is very important. So finally, we can talk about, this was number one about endocytosis. The second one is exocytosis. Exocytosis, whereby exo means we are spitting out substances which are big in size. That if I have a cell and the cell has something inside in the vesicle, and this one applies to endomembrane system, whereby substances which are produced by RAF, endoplasmic reticulum, are taken to the Golgi apparatus or the Golgi complex. The Golgi has the plan of packaging. They pack, they sort, and then they sort and transport. So packaging, sorting, and modify. After modifying, they send them to the lysosomes, whereby lysosomes are going to break them down, and the lysosomes take them to the cell membrane. And reaching on the cell membrane, they fuse with the cell membrane and they release their substances by exocytosis. And a peculiar example is receptors. The neurons, that is neurotransmitters, that if I have, this is a presynaptic knob, and this is the postsynaptic knob, that if we have in a vesicle a neurotransmitter like acetylcholine, that upon entry of calcium ions which are voltage gated, they cause a power stroke leading to the fusion or movement. So calcium ions are always released, which causes fusion with the membrane. So when this vesicle fuses with the membrane, it releases neurotransmitter in the cleft by exo. So acetylcholine is released by exocytosis. So this is a peculiar example whereby you have the neurotransmitters in the vesicle, then upon depolarization of the cell membrane due to entry of sodium ions, it causes influx of calcium, and this calcium brings about contractions within the cell, leading to this guy to fuse with the membrane, leading to release of neurotransmitter by exocytosis, hence enabling transmission of nerve impulses. So this is what we can talk about exocytosis. So thank you so much for learning with us. Hope you remember about the transport of macromolecules, whereby we are looking at endocytosis, that is taking inside the cell, or exocytosis, taking outside the cell. And we have said endocytosis is divided into phagocytosis, which is cell eating, pinocytosis, cell drinking, then receptor mediated endocytosis, and we have seen the mechanisms. Then we ended up with exocytosis, which we have seen a peculiar example how neurotransmitters like acetylcholine, epinephrine, norepinephrine, dopamine are released by exocytosis due to depolarization and entry or influx of calcium ions, which bring constructions leading to the fusion of the vesicle with the membrane, hence releasing substances by exocytosis. Hope you have enjoyed this video, and if you have learned a lot, please remember to share to many others and subscribe. Thank you so much.